we work with the Farmingham data. This is one of the sources of information for us, and they are for 60 years actually. They have 2-year measurements of the status of the health and physiological conditions and variables. So this is a very valuable source. Briefly, the outline of my talk is on the screen. I'm going to show what are longitudinal data that we are working with, why they are so interesting for a researcher. I'd like to start by saying that there are standard biostatistical approaches to the analysis of longitudinal data which we are not happy with. And at this conference we have listened to a number of beautiful talks which stressed the importance of different research projects on aging. However, I have not heard the emphasized importance mentioned of the interdisciplinary approaches. And from our analysis, I hope you would see that we think that it is very important that we have to attract not only longitudinal data, but the analysis of such longitudinal data should be based on all the knowledge which is accumulated in the area of aging, in the age-related diseases, and so on and so forth. So it all has to be interrelated. There are different trajectories for variables which describe the physiological condition. So these variables may change in a monotonic or non-monotonic way. Information about aging is actually there in such measurements, although specifically we cannot say whether or not there is a single marker of aging. Of course, there is no such marker, and that is why we have to find bits of information from different age studies which we have. And talking about the human data, I should say that we have a lot more measurements for human research rather than for experimental animals. So a human being can be a very good model to study the aging of the laboratory animals, I should say, and vice versa. And indeed, we can measure many more individual parameters, analyze
возрастные изменения у человека.
changes.
Мы увидим, что не только значения возрастных переменных имеют ассоциацию с продолжительностью жизни, но также динамические свойства. Это для нас было некоторой неожиданностью, что то, что скорость возрастных изменений, с одной стороны, должна как-то коррелировать с продолжительностью жизни, это как бы народ очень давно подозревал, но многие другие переменные, так как, например, в немонотонном случае точка достижения максимума значения физиологической переменной, а также скорость снижения этой переменной после максимума, она также влияет, также имеет связь с продолжительностью жизни. Динамические свойства физиологических переменных связаны с генетическим бэкграундом, с генетической структурой того индивидуума, для которого собираются данные. Мы тоже увидим, что наличие или отсутствие кандидатных генов, связанных для старения,
trajectories of the respective variables. Since the analysis of such data should be done with due account for the knowledge and information on aging, it has to be an integrative analysis. For instance, standard analysis of laboratory experimental animals normally would compare the curves, the mortality curve, and also the slope of that curve is usually taken to be an indicator of whether or not the change, there is a change in the rate of aging as a result of
of some exposure to a toxic or some unfavorable factor. However, such a point of view turns out to be too simplified, oversimplified. Of course, it's desirable to have such integrative model of the mortality whose parameters would reflect the behavior of the biomarkers, of the physiological variables which may characterize the process of aging. And I'm going to show that at the stage of the interdisciplinary studies, we normally start with the analysis of the data where you have to attract a model. Demographics already uses some simple models. However, the analysis of the life years requires certain mathematical efforts. And the specific feature of the longitudinal data today is that there is a lot of genetic information. For Framingham data, for instance, we know that there are 550,000 SNPs which can be analyzed together with the longitudinal data for more than 60 years now. And at present, for these 500,000 SNPs, there are some amputated data, more than 2.5 million. That is the analysis genotypic and the phenotype of the longitudinal data awaits for the specialists in genetics who might compare these data and treat them together, taking into account all the other factors. Because, of course, there are dozens of research teams in different countries. I'm going to speak about the genome-wide associated studies of human longevity, which would show how we can analyze the effect of the genes on the life expectancy and longevity. And here, an interesting longevity alleles are identified, the ones which will increase the life expectancy, and also there are alleles which have negative association. We call them vulnerability alleles. Both are present and they do correlate with each other, and I'm going to show the results of the studies of those, and I'm going to also dwell on the very important aspect of replication of the genetic studies, particularly of such which deal with the scanning of the SNPs. The problem of replication of genes which influence the lifespan is quite a challenge. The results of recent research show that many findings, many results are not confirmed for the independent populations. Some are confirmed, but some are not. Why? And from our point of view, this is due to the nature of all these, interrelations of the lifespan depending on genetic and non-genetic factors. So longitudinal data. These are data which show how we age. And they, of course, give us a possibility to summarize the effect of many factors. That's a dream of the gerontologist. But, of course, it is desirable to have those factors measured. But longitudinal data, particularly for a human being, their projection for Framingham 60 years ago when took place when many indicators were not known. It was not known what they are effective for. And also Framingham study was done to do a research into the cardiovascular diseases. But it's important that longitudinal data, particularly for a human being, and we try to understand what's happening with man. But such data will show you the long-term consequences of different interventions, of different lifestyles, and they give you a possibility to see that not all local interactions will influence the gene-to-gene interactions, but also they would show what will happen with the human being during his whole lifespan. And sometimes these changes may be absolutely unexpected. It is natural that such data actually create a potential for the integrative analysis to solve such problems using mathematical simulation, whether we want it or not. But mathematical simulation and modeling is a must if we want to obtain a systemic analysis of the problem. Okay, Framingham data. Here I describe Framingham data, and for many of you they are quite known, and there are 3 cohorts, and I'm going to speak about the results of the analysis of just one of them, which is followed up for more than 60 years. And the peculiar feature of the cohort data is in the following. We can see what's happening with individuals versus many other analyses where actually you have to have case control because you have to compare the cross-sectional analysis results, and that may lead to mistakes due to the fact that cohorts may be different and there is an effect of the population stratification. This is something which has to be controlled by different methods at every point in time, but there is no adequate method for such control to make up for all the errors, so it's non-existent. There is no such unique method, although there are many attempts to provide such a solution. These graphs, these plots show how we age. It's not just the process of aging, this is the process of changing of physiological parameters. Blood glucose. Point of this dynamic pattern. You have already heard that several sessions that there can be a non-monotonic behavior of the genetic frequencies. At genetic frequencies, the factors which may influence the lifespan. There is a deep, profound relationship between such non-monotonic changes of the physiological parameters and between the parameters at the genetic frequencies that we had already discussed earlier. So these are the trajectories which do not yet characterize the biological essence of such changes. But along with the biology, there are also structural changes. People who have excessively high pressure, they actually die out, they are dropping out. So this curve has both biological and non-biological components. How do we separate between them? In this slide you can see differences in the trajectories of the biological variables among those who had a short lifespan and those who have a long lifespan. You can see that differences are quite impressive. Statistically significant they are, and I'd like to draw your attention to the fact that this indicator, for instance, the average cholesterol or diastolic blood pressure, you can see that the curves are crossing each other, and the physiological change curves, they are not parallel to each other. It's not that one will be always higher than the other. There is such a phenomenon as crossing of the curves, and this again has a very high resemblance to what we see in the non-monotonic pattern of change at genetic frequencies. So these are the parameters. Here again you can see difference between short lifespan and long lifespan. And you can see that the differences are quite significant, and physicians, doctors have been using such phenomena for a long time already, and they can judge how much more healthier is that person compared to the other one, for instance. So the analysis of such data can give you a possibility to solve problems which couldn't be solved otherwise when using just genetic data alone, for instance. What's a physiological norm? What pressure is to be maintained at a young age? So we do have an idea of this, but looking at these trajectories, we will see that the healthiest individuals even might have change in the blood pressure in a non-monotonic way. So it is reasonable to ask, what's the norm? What's the optimum level of this indicator at different age? And the analysis of longitudinal data will answer this question. It is wise to believe that the norm is the value which would minimize the mortality at this age, and that point turns out to be different for different age. It is useful to compare not only long and short lifespan, but also healthy and unhealthy individuals. Again, you can see that differences are quite essential, and it is also important that there are again some crossings, and it's not only just one curve which is always above the other, but there are some points of crossing somewhere at the age of 70, at the age of 80. And we know from other studies that a young organism and an old organism, these are two different organisms, and they do have peculiar patterns, and it's one thing to treat a young person, and to treat an elderly person is an absolutely different task. And this is another picture which shows this crossing of the physiological curves at the crossroads, so to say, from elderly to the oldest old. It turns out to be very essential for understanding the processes which take place in our organisms, so that we understand the genetics of these processes and of what is to be done to promote health and longevity in the oldest old. And just several more slides showing that not only values of the physiological variables are used for predicting longevity and health status, but also a number of other parameters are used. And I'd like to draw your attention to the following. When the changes are not monotonic, the decrease rate after reaching the maximum is a determining factor for longevity. And there are a number of other characteristics which are also very important, such as the rate of increase to the maximum and the reduction or decline upon reaching the maximum. So these two parameters are very important. The slower is the decline of the parameter upon reaching the maximum, the longer the person lives. And also there are other parameters such as the maximum value and the age at which this maximum is reached, as well as the decline rate upon reaching the maximum and the variability. The lower is the individual variability during the life of the human being, the higher are the chances of a longer lifespan.
An illustration. The longitudinal data lets us measure these changes for each person, and having a population, or we can have a distribution of those changes, and different segments of this distribution curve can be compared, we see that there are differences in longevity of different types of people. In most cases, we see these are statistically significant differences, but they exist for all the indicators. I don't have the time to go into further details, but I may ask you questions later on. Let's look at the genetics of aging related to changes. This is not effect on the aging, but on the age changes, which are most probably related to the process of aging. The API4 gene, the dynamics is different, significantly different between the bearers and non-bearers of the gene. It was not previously studied, but looking at these dynamics, we may compare it with the dynamics of long and short lifespans. And here you see the effect of it on the body mass index and on the pressure parameters. And you see again the crossing of the curves, which says that APOE4 may be not too bad in the end of the life, and analysis of recent data shows that each of the genes has the age when it expresses itself. In some ages, it has a positive effect. At other age periods, it has a destructive effect. In this case, it seems that in some specific conditions, ApoE4 may be accumulated towards the older age, and this may be either dumped by some buffer, or there may be some other mechanisms involved. Let us now look at the non-monotonous trajectories of genetic dependencies. I worked together with an Italian group headed by Claudio Franceschi, Benedictisis, and with Vladimir Anisimov, using different sets of data. What were the outcomes? 1998 data we found with Giovanni de Benedictinis and Franceschi. We saw that there was non-monotonic age trajectories of genetic frequencies, although we previously expected that there would be monotonous growth, especially in case of frailty genes. Those who dealt with demographic studies or biodemographic studies surely says that there are crossings of the mortality curves of the bearers and non-bearers of these genes. Something happens to the motility or longevity curves here. What could be the cause of this crossing of curves? If we take the 100 age point, you will remember that since the 50s, the number of 100-year-olds has grown significantly. Why so? There were no genetic changes in the population in this brief period of time. External conditions must have changed. Some people who previously died out and did not live to the age of 100 years, in those new conditions, now could live to 100 years. This first explanation is in the change of external environment. The second explanation may be that not only external environment changes, but the internal environment changes too. Some genes are switched on or switched off during the life period, and animal experiments show that expression of many genes is really changes, and the changes of the expression set up a new genetic environment, and the gene which could be hazardous until the age of 80, after 80 years in a different gene environment created by the aging mechanism, can turn into a favorable gene and prolong the lifespan. Many habits, according to the doctor's recommendation, or the self-feeling of the person, they are changing. At some age, the person drops smoking, drinking, etc. Behavioral patterns are changing too. And this all may have an effect on longevity. You see an example of such a crossing of these curves, longevity curves, that is an illustration of this. And there are numerous examples like this. Now, Barzilai also mentioned that more than 100 such genes have been identified by now, but the explanations are not final, and they all need to be checked. These are other examples of similar phenomena of crossing. I just want to illustrate the fact that this is not a single coincidence. This is something we see in numerous cases, and this non-monotonous change in physiological variables hints that genes should be looked for here, genes responsible for those changes. Now, a few words for admirers of systemic approaches. I'll show a couple of slides to that effect. What is the model of longitudinal data that we need to develop for the age-studied research? The existing biostatistical methods of analysis of longitudinal data do not fit the tasks of an adequate interpretation of results. The longitudinal data need additional information about variables that have not been studied yet, about some regularities between those variables need to be taken into account, and a lot has been established on animal models. Here you see a list of risks that are to be studied. The mobility or mortality risk depends on different risk factors, and the shape of the curve is U or J shape. This is Hermes's effect, which is quite often seen. That is the optimum norm when there's a deviation to the right or to the left that enhances the risk. We spoke much about aging as lower resistance to stress with age. You'll not find this variable in any longitudinal data, but at the same time, this concept exists. There are indicators that have been developed at the cell level. How can we study and extract the main parameters from longitudinal data? We found a possibility for that. Changes in the adaptive capacity with age. Adaptive capacity lowers down with age, we understand that, and how can that be assessed on the basis of longitudinal data? It can be done. Effects of allostatic adaptation. What does that mean? Allostatic adaptation is a change in some physiological indicators that is necessary to support a stable level, homeostatic level. The organism, for example, may sacrifice the cholesterol level or blood pressure level to sustain the CO2 concentration in the lungs or sustain some other vital variables. Specificity of this approach is in the fact that not all stresses need to be measured, but the result of the effect of sustainable stress factors on the organism. Looking at the allostatic adaptation with age. Deviations of this curve from the norm characterize the so-called allostatic load, and there's been a lot of research to that effect, attempts to formalize this concept. This variable can be included into the model. Stochasticity. Here you see an illustration of aging. Healthy old are characterized by the upper curve, healthy young lower, and you see the effect of stress, and the sick individuals shifted along the variables, and you see that the parabola is narrower here. The deviations are changing, that is, deviations from the norm. That is the way the model describes these variables, and in case somebody has questions, I'll answer them during the break. So these parameters can be well described and assessed. I don't have time left. I want to say a few words about the rest of material. These are age changes, the model works. And a few works about the genetics of survival. Among the genes that affect survival, there are numerous genes with low effect, and the genome scan analysis very often shows these effects as non-significant. But if we study these genes cumulatively, these longevity genes, we'll see that the effect is significant, and here you see a very interesting analog between two trajectories. The left one shows changes in longevity in Australia, and the right one shows mean longevity depending on the number of longevity genes in the genome. This is illustrated by other diagrams, that is survival functions starting from 76 in Switzerland. That is the left one, and the right one, the survival function depending on the number of genes. This is a very interesting interpretation based on the saving lives model. If it is based on the assumption that when a person dies, the person may be resurrected, and the number of such possible resurrections is estimated through the notion of repairs, and that's the final result of the empirical data and its effect on the longevity function. Just 30 seconds more. The genes that we identified showed that mostly they do not affect the encoding of the protein, but they affect the volume of the protein, its concentration, its quantity. And this analysis has sense and makes it possible to continue with our studies. Longevity genes.
So these genes reduce life span, and these genes confirm a possibility of evolution hypothesis on mutational accumulation.
Этот замечательный слайд я тоже пропущу.
Conclusions. Analysis of longitudinal data is a huge resource which hasn't been used properly so far because the lab results in the re-engaging are a little bit stand a little bit apart from longitudinal data, and we need a special tool to use them. The interactive analysis is quite possible, and we need to attract interdisciplinary teams for that, to include mathematicians. So there are also longevity longevity genes and vulnerability genes effect also should be included into this study. That's it. Thank you.